With the exception of parasitic protozoa, all forms of life synthesize purine and pyrimidine nucleotides. Synthesis from amphibolic intermediates takes place at rates which are controlled according to cellular needs. Amphibolic intermediates are the compounds which are produced during catabolism of one pathway but may play part or a different role in another pathway, which in this case is the purine synthesis pathway. There are intracellular mechanisms which can sense and regulate the pool sizes or amounts of nucleotides. For example, synthesis increases during growth or tissue regeneration when the cells are rapidly dividing. Regarding the sources of the carbon atoms of purine ring, nitrogen at position 1 comes from the amino acid aspartate. Carbon atom at second position is contributed by formyl group, which comes from the amino acid tryptophan during the 1-carbon metabolism. The nitrogen atom at third position is donated by glutamine. Next, the whole of the amino acid glycine becomes incorporated during the purine synthesis, contributing carbon atoms at 4th and 5th positions and nitrogen atom sitting at the 7th position in the purine ring. Next, 6th position carbon atom comes from the carbon dioxide. Carbon atom at 8th position is contributed again by N10-formyl tetrahydrofolate. The nitrogen atom at 9th position again contributed by glutamine. So here we can see that 3 amino acids are playing part here directly. These are aspartate, glycine and glutamine. Glutamine has dual role because it contributes 2 nitrogen atoms at positions 3rd and 9th in the purine ring. And there is an amino acid which is playing part behind the scenes, contributing atoms indirectly during 1-carbon metabolism. This is tryptophan, contributing formyl groups for carbon atoms at position 2nd and 8th of the purine ring. The synthesis of purine ring starts with the sugar ribose 5-phosphate in presence of PRPP synthase and magnesium ions. ATP is converted to AMP with release of pyrophosphate. This pyrophosphate attaches to ribose 5-phosphate, resulting in formation of phosphoribosyl pyrophosphate or PRPP. Now PRPP in presence of PRPP glutamyl amidotransferase receives amide groups from glutamine. Thus the foundation of the purine ring is laid. This nitrogen atom is going to appear at position 9 of the future purine ring. During this transformation, the pyrophosphate is released and PRPP is converted to 5-phosphoribosyl amine. Next, the amino acid glycine as a whole becomes incorporated into it, contributing the carbons at 4th and 5th positions and nitrogen at 7th position of the purine ring. And 5-phosphoribosyl amine is now converted to 5-phosphoribosyl glycinamide. Then folic acid donates carbon atom, converting it to 5-phosphoribosyl formyl glycinamide. Next, glutamine donates its amide group for the future nitrogen atom, which is to occupy 3rd position in the purine ring. Thus, 5-phosphoribosyl glycinamide is now converted to 5-phosphoribosyl glycinamidine, which undergoes ring closure and cyclization to 5-phosphoribosyl aminoamidazole. So here we can see that there is ring closure. Next, the carbon dioxide contributes the carbon atom, which is to occupy the 6th position in the purine ring, leading to the formation of 5-phosphoribosyl aminoamidazole carboxylate. Next, the amino acid aspartate comes into play and transiently combines with 5-phosphoribosyl aminoimidazole carboxylate to form 5-phosphoribosyl aminoimidazole succinocarboxamide. It is going to contribute nitrogen atom at first position of the future purine ring. Next, fumarate or the carbon skeleton of aspartate is removed by the enzyme adenylosuccinase, leading to the formation of 5-phosphoribosyl aminoimidazole carboxamide, which after addition of carbon fragment for the 8th position by folic acid is converted to 5-phosphoribosyl formimocarboxamide, which is cyclized. So this is cyclized to form inosine monophosphate, which is the parent compound for the purine nucleotides. So here we can see the formation of inosine monophosphate or IMP. And this is the parent compound for the purine nucleotides, which may be adenine or guanine.